Hello, my name is Vassilis Katsigiannis, and I'm a researcher at the Institute of Communication and Computer Systems in a Biomedical Engineering Laboratory. And I'm going to present you the PHOREVER project. First of all, we will have a project overview. Then we will discuss about some technical and medical background, about the medical use cases and the medical impact of the project. Next, we will talk about how the PHOREVER workflow is and what's the project status and about some challenges and mitigation measures we took. And finally, about the impact creation. PHOREVER stands for Photonic Integrated OCT Enhanced Flow Cytometry for Cancer and Cardiovascular Diagnostics enabled by extracellular vesicles discrimination. It's funded by Horizon 2022 program, and it started about 2 years ago, and its duration is 42 months. Its budget is about 5 million euros, and the coordination is from the Institute of Communication and Computer Systems. PHOREVER aims to develop a point-of-care device using novel photonic technology in order to detect blood biomarkers for clinical application in stroke and pancreatic cancer. The consortium consists of 8 partners. 2 of them are clinical, 2 of them are related to the software development and the data analysis, and the rest of them are related to the device development and the hardware assembly. Currently we are at the 25th month of the project. So here we have some key points about the PHOREVER. As we mentioned before, it aims in developing a photonic biosensor for the detection of small size extracellular vesicles that can be used as biomarkers for pancreatic cancer and stroke. It's going to be a multi-sensing platform for automated blood sample analysis, and it integrates 2 photonic modules. The first one is the flow cytometry, which combines 2 photonic integrated circuits for the implementation of scattering pattern measurements and the fluorescence sensing. And the second one is the optical coherence tomography, which is set up as a coherent gate for the validation of the flow cytometry measurements. Finally, AI methods will be incorporated in order to develop an application-specific data analysis tool. Here are some information about the technical and medical background. First of all, what are the extracellular vesicles, or EVs? They are particles which are released by cells, and they are located within the blood plasma. These nanoparticles carrying a complement of lipids, DNA, RNA, protein, metabolites, and glycans. And its use is to serve as mediators of intracellular communication, reflecting identity and the activity of the cell of origin and carrying information about the human body conditions. Now the main module is the main technique used for measurements is the flow cytometry. It is an analytical technique used for rapid detection and the characterization of individual cells and particles in a heterogeneous population. And it provides information about the cell's form, their complexity, and intracellular structure based on the photons that are scattered and the fluorescence properties. It consists of 2 sub-techniques, let's say. First of all, we use the scattering pattern, which differentiates the cells based on their size, their structure, and their internal composition. And then there is the fluorescence sensing, which helps to detect particles of a specific kind, of a specific kind based on their fluorescence properties. Finally, the second module of the PHOREVER device is the optical coherence tomography, or OCT, which offers information about the number and the position of the plasma particles that will be illuminated inside the flow channel at each point of the time. It will be used as a mean to validate the scattering patterns from the FCM measurements and improve their resolution. Now, PHOREVER targets in two medical use cases. First of all, the pancreatic cancer, in which we aim to diagnose health patients among patients with actually healthy individuals, among patients with benign tumors, and among patients with malignant with exocrine or endocrine tumors. Then we will try to monitor the disease progression and the treatment and to assess the treatment response. The second use case is the cardiovascular one, where we try to identify stroke events among other stroke mimic conditions, such as the epilepsy, and on a second level to identify the specific stroke type among hemorrhagic stroke, ischemic stroke due to small vessel occlusion, and ischemic stroke due to large vessel occlusion. PHOREVER aims to improve the current standard of care, which mostly involves the usage of imaging techniques for the disease diagnosis, such as CT scans, MRI, or PET-CTs. However, these techniques are expensive, they are time-consuming, and in some cases, such as the CT scan, they expose the patient in the high radiation levels. So, and further, they do not fully grasp the biological mechanism that contributes to the progression, the development, and the potential resistance to treatment regimens. PHOREVER platform will try to improve the current standard of care. In pancreatic cancer, we will be able to diagnose the disease earlier and monitor its progression and the treatment response. And in the cardiovascular use case, each patient will be able to be transported directly to the optimal healthcare center according to the stroke type. Yes. And finally, this will help in eliminating the retransportations and save valuable time, which is really crucial in the case of stroke occurrence. Now the PHOREVER workflow. In the first stage is the blood sample acquisition from the clinical staff. Then this sample is processed in order to separate the blood plasma, and which will be diluted in order to be inserted into the fluorescence modules. So then the FCM and the OCT measurements take place, which will help identify the AVs among other particles in the blood plasma. Based on the AVs, we will extract some features, such as their size, their concentration, and we will also incorporate other patient information, such as the age, the gender, and the blood pressure, in order to form the final feature vector, which will be used for training and implementing the AI models. Based on this process, the AI models will serve as a clinical decision support system in order to detect the required classes. At the moment we are, since the 18th month, actually, we have some preliminary AI results based on retrospective data acquired from the project's blood sample biobank. And they are actually really promising, since in the pancreatic cancer use case, we achieved an 80% accuracy in identifying the pancreatic cancer type. And in the cardiovascular use case, we accurately diagnosed 90% of stroke incidents. And also we achieved a 70% diagnostic accuracy on the stroke type identification. Now, regarding the hardware, at the moment, some hardware prototypes are being constructed, and within the next few months, probably the next one, some tests will take place. The first medical use of the PHOREVER device is planned to be made on 36th month, and the final device on the end of the project by month 42. And after we finally develop the device, we plan to compare the results based on the PHOREVER, between the PHOREVER hardware and the current state of the art. So some challenges that we faced within the project, mostly based on, mostly related to the data sets, is that the data generation from the blood samples using the gold standard equipment were very time-consuming and expensive, which led to a relatively small amount of data. PHOREVER device will help in solving this problem in the future, since it will offer low-cost measurements. Now, also, the data acquisition from the rare groups was difficult, and there were also some features missing from different participants, which led to data miscompleteness. And there are also some technical challenges, which are mostly related to the detection of EVs with a diameter of 100 nanometers. Now, regarding the mitigation measures on the data sets, we performed oversampling techniques to alleviate the class imbalance. We seek for additional data from the clinical partners to fill some classes. And also, we suggested to the data providers which features are the most crucial ones in order to include them in a new data acquisition. All in all, PHOREVER's targets in using novel photonics technology and advanced data analytics to applicable outcomes for the healthcare sector, specifically in diagnosing and monitoring the pancreatic cancer and the diagnostic stroke types and stroke occurrences. We will deliver a portable point-of-care device at the end of the project, which will reduce the diagnosis and patient transportation time to facilitate the clinical workflow. This will also help in understanding better the diseases, mechanisms, and enhancing the monitoring capabilities. In a wider scope, PHOREVER will provide technical, biological, and many other advancements, and it will help in technology readiness level progression, propelling and transferring to medical applications. You can find more information on the PHOREVER website and all the accounts in Twitter, LinkedIn, and YouTube. Thank you very much.